Okay, well, I guess we'll get started, and I hope people will join us as the hour goes on. Welcome, everybody, to the first meeting of the working group for January 19th, 2023. I know it's been a while since we had our last meeting, so thank you for coming back. As usual, this meeting is live streamed and recorded and made publicly available. As always, Tom and I are available for comments or questions. Jim Sluka, Lorenzo, and Joel are also there helping organize, and we appreciate that. We have our various communication channels. Clearly, we need to do a better job getting the word out about the talks for this semester, and so we would appreciate your help communicating that. And we have that hashtag MSM viral that we've been trying to use to identify that. There are a couple of announcements about upcoming events. Next week, January 26th, there is a joint IMAG-MSM working group meeting. This is not our working group. This is the whole IMAG-MSM program hosted by Grace and Reed. And if people are interested in that, please let us know and we'll try to get you access to that. The other thing that is coming up that's relevant is that there is a National Academies virtual workshop, one-day virtual workshop on the 30th of January. Reinhardt is a keynote speaker for that, and Gary and I will be discussion sections for the various subcomponents as well. So there are three of us who are going to be presenting at that meeting. It looks like there may actually be a IMAG-MSM in-person conference coming up in June. Hope that happens. That would be great. And if anybody wants to look at the anniversary slides from our last meeting in December, those are available. So please feel free to take a look at those. Are there any other announcements from the community at the moment? We'll have a steering committee meeting tomorrow morning at the usual time. If you need a link and you don't have one, please let us know and we will send it to you. So that, I think, if I don't have any more announcements, I don't want to take time away from our speaker. February 2nd, we have another talk from University of Heidelberg. February 16th, we have a second talk from Nova Discovery about their multi-scale modeling package for virtual clinical trials. And then Maria Abu-Chakra on the 2nd of March is going to be talking about her very large scale digital twin initiative, human digital twin initiative, the human virtual human development project, which is, I believe, centered in Canada. It's based out of Toronto and UBC, but also has, I believe, Allen and Gates involvement and is very international. It has European participants, Japanese participants, quite impressive and rapidly growing effort. So we're glad that she's going to be coming to talk to us about that. If you have ideas about speakers, please let us know. We're always glad to add people to the roster.
If I can just add, James, if you could go back, please. So Claudio from Nova Discovery, they also offered to folks, if they want an account in their Ginkgo platform, they'd be able to set them up so that they can check it out. So if anybody's interested, just please email me and I'll facilitate the interaction with them.
Okay, I don't want to take more time away from our speaker. He's a well-known and busy man. and he's going to be talking about the large-scale immunome project that he kicked off last fall, and plans and opportunities.
Great. So if I could, how do I share the screen now? Let me get this open here. You guys able to see the slides?
Yes.
All right, great. Well, thanks a lot for the invite. It's good to be here. It's good to see a bunch of folks wherever you guys are. And I'm going to talk to you about a new idea, the Human Immunome Project, which is has been announced. We had basically had held a meeting about 3 months ago now, and launched. And the problem that we're attempting to address here is why is it that if we see an infectious disease like COVID, or we take a vaccine like HPV, or we're working in the cancer immunotherapy space, or on a range of other non-communicable diseases, that we have a bell-shaped curve of how individuals actually are reacting, and we don't understand why. We don't understand why because the human immune system is large scale, is extremely complex, and it's only recently that there has been a convergence, if you will, of the technological advances in the immunology space, systems biology space, and AI, which has given us the opportunity to even begin thinking about how do we understand or decode the human immune system. So I'm going to talk about a project that is called the Human Immunome Project, that evolved out of a large scale project, the Human Vaccines Project, and we're a non-profit research consortium. And the mission here is really to create eventually AI models of the human immune system. So if we look back over the last 2 years, we see a novel virus that emerged and spread rapidly all over the world, and has given a range of disease from asymptomatics to the mild and moderate disease to a subset of individuals, in particular the elderly, a severe disease, and a lot of mortality, frankly. But this is relatively what one is going to see across infectious diseases. And I spent a life in HIV vaccine R&D at NIH and industry and at the International AIDS Vaccine Initiative. And as we learn more and more about HIV infection, again, we saw a similar set of bell-shaped curve where a subset of individuals are exposed to HIV and basically have remained asymptomatic. The majority of people pre-antivirals had a standard about a decade-long progression to AIDS and the opportunistic infections associated with AIDS. Subset also had a rapid progression within a year or two. And there's still a subset of people who it's decades later, without antivirals, have maintained a control of this virus. Ebola is another case where we would a priori think that if you get infected with the Ebola virus, it should be fatal, and yet it's fatal in subset, about half of people. And we see a similar curve, if you will. And we're beginning to understand a little bit about the the causes and the backdrop as to why a subset of individuals are either asymptomatic or have a mild disease with Ebola. But at the bottom line, the baseline is looking like it's predictive of outcome, and we don't understand baseline. So all of us, and this is just a pictorial of the bell-shaped curve, all of us are somewhere on this curve. And ideally, we want to be in the middle. But as we well know, there's a subset of people, in particular the elderly, that as we get older and older, in the majority of us, there is a senescence of the immune system. And on the other side of the curve is the overactive immune system, which is leading to a range of autoimmune diseases, and the majority of these are in women, about 80%. So if we look outside of infectious diseases in the realm of immunotherapy of cancer, again, we only see about a quarter of the individuals are receiving the benefit. This is just an example of the influenza vaccines. Again, the elderly are doing worse, and there's a range of external factors that impact if someone's going to get a lot of immune disease and how bad that disease is going to be. And if right now we don't really have the tools, or we haven't had the tools to exploit an understanding of this. In a similar way, there is an impact of the host genetics, intrinsic factors such as age and sex, the repertoire, the B cell repertoire, B cell receptor repertoire, and the T cell receptor repertoire. But the reality is a lot of the activity is outside of the genetic sphere, and that baseline that we don't understand of the individuals, where we and others have looked and used a number of licensed vaccines in vaccine trials, and taken a lot of blood at the pre-immunization point, and look to identify one signature, another signature. A lot of these factors are outside of the realm of genetics, and some of these factors are listed here in terms of locations of cells, state of cells of the level of expression, the lymphocytes I mentioned, the VDJ and the TCR BCR repertoire, and the shifting. But at the bottom line here, we have a problem, and I think we have a solvable problem, but it's an extremely challenging one because of the scale and the complexity of the human immune system. And this is highlighted in this slide here, where the human immunome, which is a shorthand version, if you will, of the human immune system, the network of genes and proteins and cells, etc., that are the components of the human immune system, is billions of times more complex than our genome, and we're just beginning to understand our genome. But at the core, the understanding of the immune system will be the basis of our ability to be able to tackle the diseases that we haven't been able to tackle, either with vaccines or immune therapies, and some of these are listed in this slide. We have a tremendous opportunity, though, because there has been this convergence of incredible advances over the last of the decade in the biomedical sciences, which it really enable us to probe the human immune system in ways a decade ago we just were not able to do. And in addition to the immunologic assays, we have the abilities now of looking at a single cell level, and we have the whole plethora of the omics assays. at our disposal. So one of the challenges, and I think we all in the systems immunology and systems biology space are facing, is that we haven't had the capacity of analyzing the data that we're actually generating. And this is changing now, I think, because of the advent of AI. And just one example in the area of shapes and structures and foldings of proteins, it took us at least a decade, with a lot of support from NIH and the International AIDS Vaccine, where I used to work in the Neutralizing Antibody Consortium, to understand what the outer protein of HIV really looks like. In contrast, it took AlphaFold about a year to evaluate a couple 100 million proteins. So we are in a whole different era, or the beginnings of a whole different era. And certainly a year ago, the AlphaFold, the advances with AlphaFold were seen as the breakthrough of the year. And even in just a short time in the year, there are a number of other of the programs now that are built on AlphaFold, which are beginning to look at protein-protein interactions in ways that we have not been able to look at in the past. And this obviously is key when we get into workings of the B cell receptor repertoire and its ligands, and the T cell receptor repertoire and its ligands. So we decided to hold a meeting a few months ago called the Human Immunome AI Summit, where it brought in leaders in the fields of immunology and systems biology, in AI, machine learning, in big data, etc. And one of the initial things that we observed is we all speak a different language. And slowly over the couple of days, we slowly began to understand each other. And the outcome of that is the launch of the Human Immunome Project. And as I mentioned, our long-term goal, and everybody understands this is a long-term, is to begin to make AI models of the human immune system, and the output, if you will, of the meeting, which there's a paper on the desk of the editor of Nature right now, which is a summary basically of the meeting and the outputs. It is basically is a game plan, or the beginnings of a game plan on how we might approach the creation of AI models of the human immune system. So on the left on this slide here, we show the inputs, the immunologic assays, omic assays, impact of the exposome, medical history of the individual, etc. And this is either from the data that is already out there that one would have to access and curate to enable AI specialists to begin even working in this space. But eventually, if the resources are there, we should be able to initially have the preliminary models of human immunity. And just as it's taken a couple of decades in the finance area to improve their models, and in the weather and the climate models that are out there now, which were really in the preliminary stages a couple of decades ago, and have gotten a lot better. The exponential increase in power of the machines is really offering us a tremendous opportunity here, and we believe that as models are developed, these will impact how discovery is made in the biomedical space, and offers the potential of in silico vaccine trials, in silico immune therapy trials, et cetera, where the impact at the end of the day is healthier lives. So what we're really talking about here is a new initiative on the scale of the Human Genome Project, but with a level of the complexity as a result of the different kinds of the data that one has to generate, from the molecular and cellular to moving out of blood into tissues, to the individual and the population level, and to also incorporating in the exposome to enable us to begin to have mechanistic models and eventually to have causal models of human immunity. So as I mentioned earlier, I think we all understand that we're looking at a long-term effort here, and one of the major outputs of the meeting in September, which is really is the initial of a series of meetings that we're doing in that space, is to offer up a concept that of a series of what we've called the grand challenges, where the initial challenge on a topic which we still have to define could be accomplished in a 3 or 5 year window, and could point us in the direction and show enough of an impact that the ability of advancing from the philanthropy money into the major government money, which is really needed to accomplish an initiative like this, is feasible. So when we think at least of these grand challenges, what we're talking about is a topic area where the application of the advanced computing in the immunologic space can address a previously intractable problem within a reasonable amount of time, and we arbitrarily have set a 5 year window. And these grand challenges could address and be supported by existing sets of data, and or the newly the generated sets of data, and we're estimating a ballpark estimate, and it's obviously going to depend on the way that the initial challenge is defined, and projects associated with that challenge. A ballpark estimate of between 50 to 100 million over a 5 year period. So the way we've organized the project is we have an executive management team. We're in the process of setting up a scientific leadership group, which is the decision-making body for all of the scientific initiatives we're going to do. We have a board of directors, which is basically overseeing our fiscal management and giving us a tremendous amount of the strategic advice, and also opening up doors to the potential donors. But the core of the project itself is going to be a network of laboratories. We have a subset of those laboratories as carryover from the Human Vaccines Project, who will do the work in terms of the generation of the data, and or the access and analysis and the curation of the data. And then as a public-private partnership, we'll be in a position of linking in with experts in the field of AI, machine learning, et cetera. Another outcome of the meeting was this identification of the pillars, if you will, in support of the grand challenges that are shown in this slide here. And we're still working through now, because it's only been about 3 months, but we're still working through exactly, how we're going to set up each of the pillars. But in general sense, there is an infrastructure pillar, which is the research, the consortium itself. There was also a lot of interest in the potential for a pillar in the artificial or experimental immune system space. These are the organoids, the in vitro chips, etc., which would give us the possibility of the rapid evaluation, generation of some of the hypotheses. A third pillar in the TCR BCR space and identification of the ligands associated, mapping, creating an atlas and modeling off of the atlas, which also have the potential of yielding intervention leads. A major pillar in systems immunology, which is the core of what we anticipate we're going to do. And in a similar way that the innovations and the technologies are advancing, we need to be on top of that here in the project. So we'll have a pillar in terms of the new technology development. So we have not yet identified the initial challenge. I'm going to show a couple of ideas that had come up at the meeting, but yeah, where we are exactly is that we're going to let out what is an expression of interest, and make that wide open across the whole scientific community. In one of these areas initially, and then our scientific leadership group is gonna meet in the middle of the year and make a decision on the prioritization of the concepts that have come up, and exactly what that initial grand challenge is going to be. But I'll show you a couple of examples. One that came up was the idea of the atlas, of the B-cell repertoire ligand atlas, and the ability of moving from the atlas to the predictive models. I mean, right now we don't. We have a few papers and a few institutions that are working in the BCR, the TCR space, and there are some of the databases that are curated, some better than others, with accessible data. But the predictive algorithms are still relatively rudimentary in our view, and the advances with AlphaFold and RosettaFold and others now, where we can take advantage of the latest of the technologies, might really open up opportunities here. So one idea was to create a language model of the human immune system, which is based on the BCR and the TCR repertoire. But again, as I mentioned, we're still in the early stages of the canvassing of ideas, and we welcome any of the ideas in this space for what either might become an initial grand challenge or a future grand challenge. This slide here just again is showing the kinds of advances on the merging of immunology and deep learning in the cancer immunotherapies, in the cancer immunotherapy space, the recent paper in Science Advances. The other area of the focus of potential challenge, a grand challenge, that came up in La Jolla at the summit is more in the systems immunology space and the predictive of the biomarkers. And Here, the emphasis, we had a lot of discussion about the pediatric, the maternal infant space. But when we look at potential impact, we're looking at greatest impact in the populations that are most vulnerable to disease, which is the maternal, the newborn dyad. And in particular, there's a lot of work. I don't work in this space myself, but there's a lot of work around identification of the markers of the activated immune systems in the mother, and inflammatory markers that seem to be involved, or maybe a major cause of the preterm births. Another area of real interest, which was also discussed at the summit, is looking in the aging populations. The demographics of the world are changing. We're living a lot longer now, and with that is an increasing amount of immune senescence, and of course, the impacts are seen, and we saw it in COVID, and we've seen it in flu, and we see it with onsets of a number of cancers and autoimmune diseases. So are there other opportunities there in doing a real deeper dive, concentrated dive in the immune senescence space? And lastly, and this is really building on the amount of data and scale of data in the COVID arena, but the potential to improve efficacy of vaccines and immune therapies in immune compromised individuals. We are doing a vaccine trial now in HIV infected subjects in Africa, looking at a variety of the COVID vaccines as boosters, and gathering initial data in this space. And in the last couple of years, we've seen a lot of work that are looking at the biomarkers of the immune age, as opposed to the biological age and chronological age. And this again is in that potential of, if we can identify the biomarkers, can we modulate? Can we shift the curve, if you will, and slow down or impede the immune senescence that occurs in the majority of the elderly. And we're even learning through some of the single cell RNA work of different kinds of cell types now that can be identified, that again impact and fall into this category of the immune age. So our next steps really in the next 6 or 9 months is the development and getting the endorsement of the strategic plan for the project, and setting up a global call to action with the 100 or so partners that we're looking for, and in mobilizing the resources for the initial grand challenge and series of the grand challenges that are going to occur after that. In particular, we soon are going to announce our scientific leadership group. We're looking to select the initial of the grand challenge in the first half of the year, to identify the network of partners in that infrastructure pillar, and create a call to action to highlight this new initiative on the agenda of the major governments, which at the end of the day, if this is going to become a reality, it's the billions of dollars that the major governments are going to have to put up over the decades. And finally, in the philanthropy world and foundation world and the corporate world, is to ensure we have the resources to do the initial challenge. So we're on our way towards that 100 partner goal, and these are the partners that had signed on at the time of the meeting that we had in La Jolla. So I'll stop there, and happy to answer any of the questions that you all may have. Thank you.
Thank you very much. That was a very helpful introduction. I think it's fascinating seeing your perspective on this, because it's almost completely orthogonal to the ones that we've been taking. And I think that the key reason for that is timescales. You're looking for prognostics, and you're looking for long time prognostics. And while some of our discussions here have been in that area, mostly I think the expertise that we've developed has been in short-term, rapid changes of immune state. And so our focus has been on questions perhaps like, if you're in septic shock, what can you do to save the patient? How do you intervene? We've had some people talk about adjuvants. Molecular recognition has not been something we've talked about a lot. But the flip side of that is something we would be very concerned about, which maybe comes under your new technologies, is how do you actually measure cytokine levels in an individual in real time? If we were going to try to say what would the key enabling technology for doing predictive modeling of immune state for humans would be, it would be to be able to do non-invasive cytokine panels. And to me, that's one of the great challenges. And so I think it's very interesting, because you're identifying very important problems, which I think everyone would agree are critical, and very interesting strategies for solving them. There's a whole, in a sense, we're coming from such different directions. It's very interesting to try to think about points of contact between those two points.
Yeah, no, I think it's a great point that you make. I mean, the ability of measuring cytokines in real time is a challenge. We at HIP just invested a little bit of money in a biotech company that really isn't off the ground yet, but has a number of the protein array platforms as microarrays, and you can look at the whole virome or bacteriome, what has been done is to look at the 100 different isolates of the influenza viruses over the last 100 years and such. But in theory, one should be able to look at cytokines, and frankly, the new burgeoning area, if you will, of the antibodies against cytokines is some of the interferon work in severe COVID cases has shown. But the key is to how to do that, and how to miniaturize the assays, and what is feasible out of a micro drop of blood, or a blood spot, and what isn't feasible. Without that, we've run vaccine trials where we've done inpatient studies, and looked an hour and 3 hours and 5 hours after immunization, and went into marrow and went into the axillary nodes, and gathered up a lot of samples. It was those kinds of studies eventually that convinced us that we needed the AI experts to help us out, because we couldn't analyze the data. But I take your point, and I'm sure that there are multiple areas of the connectivity between individuals looking at short term and individuals looking at longer term. And it's an interesting idea.
Wayne, I think so. If I remember correctly, some of the final days discussions also included some voices, including myself, but also Shai and Jacques mentioned the need for the mechanistic modeling aspects in combination with AI. Has that been sort of incorporated into the conceptualized architecture of the human?
Yeah, I think so. I mean, I kind of brushed over it pretty rapidly, but the pillar two with the organoids and the artificial immune system, there was a lot of discussion, as you know, about what can the animal models and which animal models assist us here, in understanding what we want to understand. And we just haven't been able to flesh that out yet. I mean, I think there's a lot of work. We just got some money from a foundation to begin landscape analyses on questions that we're asking around what is a grand challenge, and I think hopefully this information is going to come out over time, Tom, because I think the way we're thinking about it, and we're not quite there yet, is to create a scientific leadership group, a subset of which would be overseeing each pillar. And getting a cross section of people. I mean, it's probably better to give a talk like this a year from now when we're actually, have set up and actually are doing a grand challenge, and we can be more specific on the topic. But, we are where we are. We're building the ship and sailing as we go. And so I think, the intent is to take that information that came out of the meeting, and flesh that into an organization. The worry that I have, and I voiced this to a number of people, including Shai and John Sang and Stuart Russell and others who we've been working with a little more since the meeting, is that if we had a different set of 50 or 60 people at that meeting, we might have had a whole series of different outcomes. I mean, the whole spectrum of immunology is so great that we had people in the room who pushed one idea or another idea. So we have this, and I don't know if we're going to do this, but I was talking with the team earlier today about we have the Immunome report, which is a report that comes out every couple of weeks or so in the immunology AI space now, and I thought of, or somebody on the team thought of doing a canvassing of the field, and the Immunome report's reaching about 15,000 people or so. So we think we have a pretty wide audience, and there might be a better idea under a rock out there that we don't know about. So we're trying to be as transparent and inclusive as we can, and we're the first ones to say we don't have all the answers to what we're trying to do yet. So if you guys have ideas, you know how to reach me, and we'd be happy to entertain them.
Thank you. So there is a question by Slim in the chat. Slim, would you like to ask the question?
Sure. Just sometimes my microphone doesn't work so well. So great talk. I was just thinking when you were showing the different omics that you're thinking of using to feed the AI model that you're going to build to answer one of those challenges, I was wondering if you're prioritizing more assembling diverse data going across different assays, or you're mostly favoring a large amount of data, but for a few of those technology as a proof of concept, of course.
Yeah, that's a great question. I think it's going to depend on what the question is that we're going to ask. We've done it each way in the work in the Human Vaccines Project for the last half a dozen years. As you layer on more and more of those omics assays, none of those assays are validated, and which is is a worry for the overall field. I mean, we're getting a lot closer on. I'm not going to speak, and and a couple of other assays. But if you do metabolomics assays across different labs, you're likely to get different answers. And so that's one of our challenges. So I don't have the answer for that until we have selected, a scientific challenge. If, for example, if the first challenge is to get the whole repertoire, the B cell receptor and T cell receptor repertoire in X thousands of people across the world, where there's a diversity of age and gender and ethnicity and etc., then most of that data is basically is going to be the sequencing data, and so that's easier to curate, and that's easier to analyze. If, on the other hand, if we want to look at predictive markers in the vaccine space, the better data that I've seen in the recent papers that have come out across 11 vaccines or 13 vaccines and such is the ability to integrate data across the different assays with the programs like the Network Analyst and Diablo and others would probably be a preferred method.
Thanks.
Winnie, thank you for a great talk. This is Chang from Emory, Environmental Health. I think at the beginning of your talk, you were mentioning this kind of big variability between individuals, right? In response to vaccine, pathogen, whatever. And you're talking about that genetics probably only explains a small part of this variability. And as with people in environmental health, we tend to think that's probably environmental exposure differences can probably explain 80% of individual differences. So I saw in your one of your slides you have exposome units. So have you given any thought how you actually incorporate these exposure differences in this whole overall project? Mm-hmm.
That's a great question, and the answer, the truthful answer is we have to get the best and the brightest to work in the exposome space to be able to, to tell us how to do that. I mean, the old studies that of Mark Davis in twins convinced me that 85–90% of what's going on is outside of genetics, and as you well know, working in the environmental health space, if you're living in in India right now, in Delhi, you've got all kinds of issues on your macrophages in the lung because of what you're breathing in. If you're in the desert, you've got the immune suppression from the UV light. I mean, there are all kinds of factors, and somehow there has to be an algorithm or prioritization of ways of looking. But again, it's going to depend on what the initial challenge is, what the topical area is, that we're going to look at. I think you make an important point, though, that whatever we end up looking at is going to be across a variety of types of individuals across the world who would have been exposed, or whose HLA types are different, or I used to do a lot of work in Africa and India, and the infections that they're exposed to, the parasitic infections, for example, are skewing the immune system to a Th2, etc. So we've got a lot of issues. We can't take them all on at the same time. We got to walk before we're going to run, but we'll get there.
All right, thanks.
Wayne, I know you have to leave in 2 minutes. I was wondering, you mentioned the Human Immunome report or newsletter. Is that what you said?
Yeah. We used to put out a COVID report for a couple of years, trying to synthesize the data and whatever. But if you go to our website, there's information about the Immunome report and about the webinars that we do. Also, we do a monthly webinar at the interface of immunology and AI, and so you can find all that information. You can go ahead and register there. If you have any trouble at all, just go ahead and send me a note, and I'll point you in the right direction of the right person who can sign you up.
Okay, thank you. Thank you.
Yeah. And frankly, if there's anybody in your own group, across INAG, is that what it's called? I guess, who you think would be good, as a speaker in the Immunome webinar that we have. We reach a lot of people, and so keep that in mind too. So I do have to run. I'm sorry, but I've got a hard stop. But appreciate the time, and again, if you guys have any ideas that are gonna help move the needle, that you know where to find me now.
Okay, thank you so much, Wayne. Really appreciate your time.
All right, take care.
Thank you.
Bye-bye.
Bye-bye.